So next we'll have Jivan Khlghatyan from the Beaulieu lab at the Université Laval. And he'll be talking today about the functional interrogation of mood-associated gene interaction using CRISPR-Cas9.
Thank you. I just want to thank organizers for a great conference. It's my third time in a row I'm here. And thanks for the opportunity to present my work this year. So let's talk about using Cas9 to study mental disorders. Mental disorders are one of the leading causes of disability and a big economic burden for the society. But we still don't quite fully understand the origin of the disease. One way we look at it is the genome-wide association studies, which basically identify genetic risk factors linked to the disease. One example is the recent biggest GWAS that identified 108 schizophrenia-associated loci. But most of the time, each loci has a very small effect because of multigenic nature of the disease. So for us, it makes sense to study gene-gene interactions. We think it will be very beneficial. But how do we study these interactions? One way that we undertook with our collaborators, we identified certain genetic variants that are linked to differential expression of the mRNAs in human dorsolateral prefrontal cortex. And we found interaction between two genes. One gene was FXR1. It's an RNA-binding protein, and it was in the list of that 108 risk factors for schizophrenia. The second gene is GSK3 beta. It was not in the list of those 108, but interestingly, this gene is a target of mood stabilizers like lithium and target of several antipsychotic treatments. And we found functional interaction between these two genes, and it would regulate emotional stability in human. And interaction was in a way that people who were expressing higher levels of FXR1, they were more emotionally stable only when they had normal or low expression of GSK3. And when they had high expression of GSK3, the effect was gone. They were not emotionally stable. This was done in control patients. Later, with other collaborators, we've shown the same interaction between same variants in bipolar patients, and in this case, it was associated with symptom severity. This is all exciting and great, but we still don't understand how this interaction works functionally in the brain, what it does. And here, to understand it, we turn to mouse models, and this is where CRISPR technique comes very powerful and handy. So we wanted to induce knockouts of the genes, somatic knockouts in vivo inside of adult mouse brain. And we used dual AAV delivery system that was introduced before. So we encoded Cas9 in one AAV virus and the guide and GFP in the second one. The system was developed in Dr. Zhang's lab and was kindly provided to us. And the way to overexpress genes, we just simply used the GFP tagged genes and delivered again by AAV viruses. All the viruses were injected in the medial prefrontal cortex of mice with appropriate controls, of course, and 3 weeks after, various tests were undertaken. The first thing we looked is whether we could efficiently induce the knockouts. So, for example, in this case for GSK3, we performed immunostaining and quantified the presence of the protein. And out of all the cells that were infected by the viruses, we found that 63% of them were devoid of GSK3 protein. This was quite a high percentage for us. And to detect the overexpressed protein, we just simply look by Western blot to detect GFP-tagged protein bands. Next, we performed various behavioral studies in mice that were lacking GSK3 or overexpressing FXR1. In this case, we performed anxiety-related tests, and we find that these modifications, where the mice overexpress FXR1 or are devoid of GSK3, those mice had less anxiety. Or another word to say is that they were more emotionally stable. This was very well correlating with human studies. Next, we wanted to understand what is happening with the neurons in the brain. So we decided to patch those neurons and record the electrical activity. And what we have found is that GSK3 knockout mice and FXR1P overexpressing mice had decreased spontaneous activity, we wondered whether this was the reason of the changes in the behavior. And we think yes, because when we try to mimic this decrease in activity chemigenetically by silencing neurons, we could recapitulate the same anxiolytic type of behaviors. The next point we were interested is that what kind of neuronal plasticity can be regulated by these FXR1 GSK3 interactions. And to cut the long story short, we found that a specific type of neuronal plasticity called homeostatic-synaptic plasticity is regulated by this interaction. And we tested it in vitro and in vivo models. For in vitro model, we used a model that is called synaptic upscaling. This is basically cortical neurons in a dish, in a culture. And when you subject them to long-term activity deprivation, for example, for 2 days, and they start to compensate and bring their synaptic activity back by strengthening their synapses and increasing the activity. And this could be easily detected by, again, simply patching the cells, recording the activity, and comparing to controls. So first thing, we noted that when we do these changes, we note the expression of one of the protein of interest, FXR1, changes. So we wanted to understand, is this just a consequence or it has an important role? And we again turn to CRISPR system here now to knock out genes in vitro in neurons. And when we were recording from controls, we saw that this great 150% increase of the neural activity that was reported. However, when we knock out GSK3 or overexpress FXR1 in those neurons, this activity increase was completely buffered and blocked. Next, when we induced FXR1 knockout, we saw that already in control condition, increase was evident. And activity deprivation didn't further increase the activity, meaning that FX was actually one of the key players in this process. And when we induced double knockouts of GSK3 and FXR1, we found that GSK3 knockout effect was completely abolished, meaning that FXR1 was truly acting downstream of GSK3. So, but this could be simply just an in vitro phenomenon of neurons in a dish. So we wanted to achieve the same thing in vivo in the mouse brain. And model we looked for that, it's called a sleep deprivation model, sleep deprivation induced plasticity. And the idea is very simple. And it's conserved throughout all the animals, from Drosophila to human. Basically, when we are awake during the day, our synapses get strengthened. And when we go to sleep in the night, the synapses get weakened, and so on and so forth. However, at the end of the day, if you don't sleep, or if you block the animal or sleep deprive the animal, activity will continue to rise. And this is where it's a nice window when you can compare the animal who didn't sleep with the animal who sleep exactly at the same time by recording their neuronal activity, and you can see the difference. The animal that didn't sleep will have higher neuronal activity. And that's what actually I've done. I just sleep deprived mice and myself to study this phenomenon. And again, while doing this, we saw that FXR1 levels would change the similar way that they were changing in our previous in vitro model. And we thought that, okay, let's see if overexpressing FXR1 or blocking or knocking out GSK3 can block this increase. And that's exactly what we have found. In controls, the mice that didn't sleep had this significant increase in their neural activity, but our modifications would completely block and buffer this increase. And interestingly, many mental disorders come with sleep disturbances. And sleep disturbances or sleep deprivation themselves have deleterious effects on mood. So we think that this pathway is linking all these phenomena together. We still have lots of studies to do, but I think we are discovering something very interesting here. Just to summarize, I showed that an interaction of GSK3 and FXR1P regulates synaptic strength via most probably regulating AMPA receptors. Come to my poster tomorrow for the details. There is much more things I can show. And what is very interesting, all this is done in response of environmental stimuli. This system can buffer the changes and give the output to behavior to control mouse behavior and we think human behavior as well. Just want to thank the people who were involved, my supervisor, Dr. Martin Beaulieu, and students that work with me in the lab, Alessia, Alexandra, and Arash, and the collaborators who were involved in the study. Thank you very much.
Do you have any questions?
Quick question. I think what's going to be powerful or maybe practically very difficult is to engineer the variant instead of knocking out the overexpression of your two genes.
Yes, thanks for the question. The question is about, wouldn't it be more powerful to engineer the variants, the exact the changes that we got in the human genome instead of knocking out or overexpress? Yes, of course. That would be very precise, because here we either completely abolish or overexpress it too much. But the thing is that these variants were identified in human, and we are not sure that the same variants would result in the same phenotype in mice, and that these variants were used as a proxy of human having less expression or more expression of the genes to find the correlation with behavior. But in mouse, we wanted to understand what's the functional role of this interaction. Yes, it's drastic, but it gives an idea what these models are doing. But yeah, it will be much better to engineer the variants if we are sure that they are doing the same thing in mice. So it's hard to model the human disease in mice.